Okay guys, if you are in Advanced Bio 2, this is your chance to review with me cellular respiration. Now if you can remember that flow chart that we made on the board in class, we want to break cellular respiration down into two different options. Remember that we always start with glycolysis. So glycolysis is our first step no matter what kind of respiration we're going to do. The second step will be determined by whether or not there is oxygen present in the cell. So we can have oxygen there on the left and no oxygen here on the right. Now we'll start here on the right. If the cell does not have oxygen, it will move from glycolysis into fermentation. And remember, from fermentation, we are just trying to regenerate NAD+, so that we can go back and start over with glycolysis again. And remember, this was called anaerobic respiration. Now the other side, if you do have oxygen, we call it aerobic respiration. And we can move from glycolysis secondly into the Krebs cycle, and third into the electron transport chain. So let's take a look at aerobic respiration step by step, starting with glycolysis, and then moving into the Krebs cycle, and then wrapping up with the electron transport chain. So the first step is glycolysis. If you listen real close, you might be able to hear Charlotte screaming in the background right now, but that's okay. So when we start with glycolysis, we first have glucose. Right, glucose is our substrate that we always start with. And glucose has 6 carbons. Now the whole point of glycolysis is essentially to split glucose into 2 3-carbon molecules that we called pyruvate. So we start by investing 2 ATP. So we got to burn 2 ATP to get this whole thing started. But by doing that reaction, we are able to split glucose into 2 pyruvic acids. For just a moment, it will be called glucose phosphate. But that will be an unstable molecule that we can then break into 2 pyruvic acids. Then, the next step will convert pyruvic acid into pyruvate, which is what we're hoping to generate by the end of glycolysis. Now the nice part about this second step here is that we're going to make a couple things. Right? We're going to be able to make 2 ATP for each of these pyruvates. Oops. And we're also going to be able to make 2 NADH. I take that back. Sorry, it's just 1 NADH for each of those. So 1 NADH. So, for glycolysis, our net totals are going to be 2 ATP, because even though we made 4 down here, remember we spent 2 to begin with. So we're going to end up with 2 ATP, and we're going to end up with 2 NADH for glycolysis. Now, that's just the first stage of aerobic respiration. The second stage is the Krebs cycle. So let's follow one of these pyruvates into the Krebs cycle. See what happens there. Okay, so to begin the Krebs cycle, you're going to start with the pyruvate that we formed back in glycolysis. And remember that has 3 carbons. And pyruvate can enter the mitochondrion. So it's going to go to the matrix of the mitochondrion where the Krebs cycle takes place, but it can't enter the real cycle portion of the Krebs cycle yet. First we have to do what we call the preparatory reaction. And we're going to turn pyruvate into a 2-carbon molecule called acetate. Now to do that, we're going to have to get rid of a carbon. The way we're going to do that is as carbon dioxide. Alright, so remember, the Krebs cycle, the purpose here is to turn this pyruvate, basically all the carbons from carbohydrates, into carbon dioxide. And so there's our first one that we're going to be able to make. Now acetate also still cannot quite enter the cyclic portion of the Krebs cycle. It first has to be turned into a molecule called acetyl-CoA. And the way that we can do that is we can combine acetate with something called coenzyme A. So acetate plus coenzyme A is going to yield acetyl-CoA for us. And this is actually also going to make an NADH at this point. So we're going to make another electron carrier at this stage in the Krebs cycle. Now, acetyl-CoA, which still has 2 carbons, is able to enter the cyclic part of the Krebs cycle. And the way that it will do that is it will join with a 4-carbon molecule called oxaloacetate. Oxaloacetate. And that has 4 carbons. Now, once it does this, this is going to generate a 6-carbon molecule called citric acid. And remember, that's the other name for this cycle, so the Krebs cycle or citric acid cycle. Now from here, it's really nice, because there are several steps that happen to make this a cycle, but you don't have to know those. You don't have to know the specific enzymes or the intermediates that we make as we go along here. All you have to know is that several steps occur to allow us to regenerate oxaloacetate. Remember, this is a cycle, so we have to be able to remake whatever we started with so it can happen again. So once we've gone with citric acid, we're going to convert it several times until it turns back into oxaloacetate, which we can combine with a new acetyl-CoA, and we can do this all over again. Now, what you do have to know is that in all these stages, we're going to make a couple of different things. The first thing is that we are going to get the other 2 carbon dioxides out. So we're going to form 2 carbon dioxides. We're also going to form 3 NADH when we do this, somewhere along here. We're going to form 1 FADH2, which remember is just another electron carrier, just like NADH, that can take stuff to the electron transport chain. And we're going to form 1 ATP. Okay, so to take a look then at the payout for Krebs cycle, for every glucose, which remember means 2 of these pyruvates, we are going to get 2 ATP. We are going to get 8 NADH. And we are going to get 2 FADH2s. So this makes a little more stuff than glycolysis did. Alright, so that's the second stage. Now we want to move on and look at the third part, the electron transport. Now remember, at this point, our carbons are gone. Right? Our sugar is gone. And so all we have left are our electron carriers. We've got NADHs that came from the Krebs cycle and glycolysis, and we've got FADH2s, which came from glycolysis. And what they're going to do, that came from the Krebs cycle. And these are going to bring their electrons to the electron transport chain, which remember is a series of 4 cytochrome complexes that are lodged in the inner mitochondrial membrane. So we've got here our inner mitochondrial membrane. That means you've got the matrix on one side, the very inside part of the mitochondria, and you've got the intermembrane space here on the other side. Now remember, these complexes are just numbers. So we got complex 1, we've got complex 2, we've got complex 3, and we've got complex 4. The electrons from NADH and from FADH2 are going to be dropped off in a complex, and then they are going to be passed from one complex to the next. The electrons are going to move from complex 1 to complex 2 to complex 3 and to complex 4. Now complex 4 is then going to hand them off to a very important thing, and that is oxygen. So the oxygen becomes our final electron acceptor, takes these electrons and is able to form water as a byproduct or a waste. Now that's all great, but the thing is, we've seen what happens to our electrons, but we haven't seen the production of ATP. So how are we going to make ATP? Well, remember that as all of these complexes are passing their electrons down, they are going to be pumping hydrogen out of the matrix. And this is a pump, right? So this is active transport, pushing these hydrogens out against their gradient. So we're building up a hydrogen gradient that is greater here on the outside in the inner membrane space. The idea here is that we'll build up this gradient so those hydrogens will want to go back into the matrix by diffusion. And the way that they're going to do that is by passing through an enzyme called ATP Synthase. Now, ATP Synthase is also lodged in the inner mitochondrial membrane, and as the hydrogens move back through this ATP synthase, it is going to power the production of ATP, and lots of it. So as those hydrogens move back down their gradient into the matrix, ATP synthase is going to be able to form a whole bunch of ATP. Now how much? Right? Remember that 1 NADH is going to be able to give us 3 ATP, and 1 FADH2 is going to be able to give us 2 ATP. So let's take a look at one last slide and total all this up. For aerobic respiration, we are going to get per glucose we're going to get 2 ATP from glycolysis. We're going to get 2 ATP from the Krebs cycle, and we're going to get 34 ATP from the electron transport chain, for a grand total of 38 ATP for every glucose that goes into aerobic respiration. Now remember, there's a lot more stuff on your test that could come into play, but pretty much this is everything that you need to know to be confident going into this test. If you could get close to doing what we just did on these slides, you will be able to answer pretty much any question that I would give to you. I know this is a complicated process, but if you look at it enough times, I think you'll gather the information that you need to do really well on this test. I hope this helped, and I will see you soon. Good luck.